Pronto. Ok. Bom dia. Good morning. Queria agradecer a presença de vocês. Vamos dar início à nossa reunião científica. Essa é a primeira reunião científica do Laboratório de Pesquisas sobre Psicanálise com Evidências e Perinatalidade, o PEP Lab. Este projeto, ele nasce de uma parceria minha com o Rogério, e nós propusemos à USP um projeto de inovação, pesquisa de inovação. E esse projeto foi muito bem recebido pela universidade e nós tivemos então o direito a contratar um professor. O processo seletivo já foi feito, a professora Elizabeth Shepard está em fase de finalização, tomar posse já desse posto. E nós começamos, então, esse novo projeto de pesquisa, onde nós vamos introduzir no IPUSP uma linha de pesquisa e uma possibilidade de pesquisa que está sendo iniciada por Rogério, mas uma parceria muito grande entre a psicanálise e as neurociências, né? Então, estamos nos aprofundando nessa direção. E hoje nós temos, então, o nosso primeiro encontro, cujo título é Contribuições das neurociências para as pesquisas sobre desenvolvimento infantil, né? Eu sou Leopoldo Fulgêncio, do meu lado vocês já conhecem, temos o Rogério Lerner, e temos como convidados hoje o professor Daniel Fattori, pesquisador do Laboratório de Psicopatologia Terapêutica Psiquiátrica do Instituto de Psiquiatria do Hospital das Clínicas da Faculdade de Medicina de São Paulo, e a professora Vitória Leong, professora do Departamento de Neurociência Cognitiva e diretora do... Como é que traduz aqui, Rogério?
Ela é diretora do Centro de Pesquisa em Potencial Mental Precoce e de Bem-Estar na Universidade NTU de Singapura.
Então, eu gostaria de agradecer muito a Daniel e a professora Vitória por estarem aqui, e eu passo a palavra para Rogério, para que ele possa também fazer algumas considerações iniciais e chamar Daniel e Vitória para a mesa.
Só vou complementar muito brevemente o que o Leopoldo já apresentou. O concurso que levou à contratação da professora Elizabeth Shepard foi um concurso obtido numa concorrência aberta para docentes de toda a universidade e que disputaram entre si, pelos seus projetos acadêmicos, 63 novos claros de docentes para trabalharem nos projetos vencedores, né? Então, foi uma surpresa, uma enorme alegria para a gente, né, ter tido a honra do nosso projeto ser selecionado. Acho que foi o único aqui da psicologia. Não sei se na psiquiatria teve algum também, Daniel. Até onde eu soube, não, né? E, bom, num certo sentido, é uma vitória da psiquiatria também, porque eu tenho vínculo subsidiário com a psiquiatria e muitos anos de pesquisa em colaboração. E eu já conhecia a professora Elizabeth de muitos anos, sempre tive muita admiração, não só pelo trabalho dela, ela é uma neurocientista muito qualificada, vinda do Reino Unido para o Brasil e trazendo muito do seu conhecimento, da sua experiência e das suas conexões acadêmicas, qualificando a área de pesquisa de desenvolvimento infantil, eletrofisiologia, neurociências aqui no Brasil. E, além de tudo, é uma pessoa de uma gentileza única, uma pessoa muito agradável de se trabalhar, muito leve. E quando então ela passou no concurso, isso me fez pessoalmente muito feliz. Além de toda a perspectiva de trabalho conjunto, e ela desde o primeiro momento tem se mostrado muito aberta, muito parceira, e ela já tem uma colaboração com a professora Victoria de longo prazo, e eu acho que a gente pode falar, né, que, além de toda a colaboração do Instituto de Psiquiatria, né, nas pessoas do professor Guilherme Polanczyk, o Daniel Fattori, que está aqui, e toda a equipe, Adriana Argeu, Monique, né, Daniel? A quem a gente é profundamente agradecido, a esse vínculo de amizade da Elizabeth com a Victoria, também de longa data, de parceria. A gente também tem uma enorme gratidão por isso, um reconhecimento. Era para a professora Elizabeth estar aqui, então eu quero notar que ela está, de alguma maneira, e por algumas questões não foi possível que ela estivesse aqui hoje, mas eu gostaria de deixar registrada a nossa admiração, nosso agradecimento e o nosso carinho também pela professora Elizabeth Shepard. E, bom, com isso, eu vou dar início às atividades de hoje. Eu só gostaria de... Você já apresentou? Eu apresento aqui, tá. Então, eu vou falar um pouquinho mais sobre Daniel e professora Victoria. Mas antes eu queria saber o seguinte: todo mundo aqui entende inglês? Alguém tem dificuldade de entender inglês? Não. Ótimo. Então, não, às vezes as pessoas falam. E muitas vezes falam, não, eu quero que você traduza. Eu acho que pela Vitória, né? Ok. Muito bem, então, finalmente, a professora Victoria Leong investiga sincronia neural entre mães e bebês, integrando neurociência, psicologia, linguística e modelagem computacional. Ela ganhou o prêmio Robert Glushko, da Cognitive Science Society, de 2014, tem uma nomeação dupla, tanto na Nanyang Technological University e na Universidade de Cambridge, onde é chefe do laboratório Baby Link, do Departamento de Psicologia. E ela também tem uma bolsa de pesquisa em Ciências Sociais e Humanidades, de 2020, pelo Social Science Research Council. O professor Daniel Fattori... Fattori ou Fattori? Fattori, desculpem. Bom, mas posso fazer uma piadinha boba? A ordem dos fatores não altera o produto. Aí, no caso, Daniel é genial sendo chamado de Fattori ou Fattori, para perdoar minha ignorância. E ele é doutor em psiquiatria pela Faculdade de Medicina da USP, orientador nesse programa de pós-graduação, investiga a saúde mental com ênfase em estudos epidemiológicos, ensaios clínicos e implementação de soluções digitais para coleta de dados e intervenções. É pesquisador do Laboratório de Psicopatologia e Terapêutica Psiquiátrica do Hospital das Clínicas da Faculdade de Medicina da USP. E é coordenador científico da Corte de Neurodesenvolvimento Infantil, Germina, cujo pesquisador responsável é o professor Guilherme Polanczyk. Tem uma série de outros pesquisadores, tanto na posição de pesquisadores principais como de associados. É um estudo multicêntrico, internacional. A USP montou uma equipe oficial sob a liderança do professor Guilherme e eu tenho a honra de ser um pesquisador associado nesse grupo. Então, com isso, eu vou pedir que o Daniel venha começar a apresentação dele aqui, tá bom?
Alô? Tá funcionando. Vai.
Eu sabia que eu estava esquecendo alguma coisa. Eu espero que muito em breve a gente tenha o Daniel como professor aqui no Instituto de Psicologia. Então, eu tenho pessoalmente incentivado que ele preste o concurso aqui, para que a gente tenha como colega docente desse instituto. Então, eu queria deixar isso registrado. Vai ser um ganho muito grande para nós, tanto profissionalmente como pessoalmente.
Obrigado. I'll start in Portuguese just a little bit. Obrigado, pessoal, por estar aqui. Obrigado, Rogério e Leopoldo. Fico contente com o seu comentário agora. Até brinquei outro dia com você, né? Sempre me meti muito com os médicos, apesar de ser formado em psicologia, e agora meio que voltando às origens, né? Voltando para cá, assim, vindo para falar aqui no Instituto de Psicologia. Então, fico bem feliz mesmo de falar com colegas e pares. Então, eu vou virar a chavinha para falar inglês, para a Vicky poder acompanhar também. Today I'll be talking very briefly about our study that Rogério already mentioned briefly. It's the Germina cohort. So let me just... Okay. So I'll give a very, very brief introduction, just for the sake of time. So for the past 100 years or so, we scientists in health sciences are very much interested in understanding the diseases and mental disorders in terms of causality. We want to understand how these diseases and disorders come together, how they happen, how they affect individuals, and even though we discovered a lot in the past centuries or so, we still have a lot to learn. We made great strides in terms of infectious diseases and similar diseases. But in terms of mental disorders and some diseases, we still have a lot to learn. And in the past decades or so, we have been using this early life origins perspective to understand not only diseases, but also mental disorders. So we have to look at this picture to actually understand a little bit how behavior, symptoms, mental disorders come and actually happen in communities, individuals, societies. So we have to go back very, very early in life, even before life. Also, we have this transgenerational perspective nowadays that we assess families and then assess their babies and then the babies of the babies and so on and so on. So this is very much important. And also it's very important to try to understand every potential perspective that we can. So as you can see here, we need to understand the brain, we need to understand behavior, symptoms, but we all need to understand the genetics, the environment, and also at the molecular level. So if we don't embrace this complexity, we will not understand how these mental disorders and disease happen in humans. So we need to embrace these complexities, assessing individuals across time and considering all these potential data modalities. And then our major goal is to piece together this huge puzzle, very complex puzzle, piece by piece, to understand how mental disorders and all other sorts of behavioral problems appear. So for this, because of this, we have the Germina study. It's a very big project that, as Rogério mentioned before, is led by Professor Guilherme Polanczyk at the Department of Psychiatry in the University of São Paulo Medical School. This is part of the Welcome Leap 1KD program, and that's how we met Vicky at first. She's also part of this program. So they were very generous in terms of funding, especially for Brazilian standards. So we got funding to run this cohort and also to collect, process, and analyze a series of biological data that I will show a little bit later in this presentation. And since this project is very big and very complex, we needed help from a lot of people and a lot of departments here at the University of São Paulo. So we had help from not only the medical school, but genetics department. We have people from the microbiome department. So it's very complex and big. We have a lot of collaborators, and of course also Rogério and Lizzie in this project. So the main goal of our cohort is to develop a fully integrated model of developmental data in the first years of life to predict executive functions at 3 years of age. This is our main goal in terms of the 1KD program, but we are not ending. We are not only focused on executive functions. We have enough data to try to predict all sorts of outcomes, behavioral outcomes, cognitive outcomes, early signs of mental disorders, and all other sorts of outcomes. But for the purpose of the 1KD program, we are focused on executive functions, okay? So I will talk a little bit about the methods, and I will not be able to mention every data, every variable, every characteristic, every outcome that we have, but I'll try my best to mention the broad domains that we have. So the first thing is that we have a prospective cohort. So the idea is to follow mother-infant diets across time. So we start at 3 months of age, so very early in life, as you can see. And then our idea is to follow them up to 36 months of age. In between our baseline and our endpoint here, we have T2, T3, and T4. And we have this sort of advanced, a little bit complex cohort design that groups of children are followed at different ages, so that we can collect data across all months, all age points, without having to analyze them every month. So so we are trying to cover all the age range in between baseline and endpoint here. Okay, so our data is being collected at the Institute of Psychiatry, and we also had a lab at Educandário Dom Duarte. We don't have this lab anymore, so all the data collection is happening up to our Institute of Psychiatry, and just to mention a little bit how the data collection goes. So each time point that I showed here, we have 2 days of assessment. So from baseline to endpoint, the family will have all the families have an online assessment. So it usually goes around 3 to 4 hours, maybe more, maybe less, depending on the family. So we cover all questionnaires, scales, risk factors, and stuff like that. And then they also come to our lab. So in our lab, we have a variety of assessments. We collect biological data like saliva and feces to analyze the gut microbiome. And we also conduct developmental assessment using the Bailey scales and also EEG. So it's a very, very intensive data collection. I would say we are very, the families are very generous with us, I would say, because they have to come, they have to answer so many questions. At each time point, they probably have like 4 to 8 hours of assessment if we sum all together. So they are very generous with us, and I would like to thank every family that's been with us since the beginning of the project. And we have a very low dropout rate too. This is very, very interesting. So usually these families stay with us for this very long time. So this is just some pictures of our babies at the baseline doing the EEG. So as you can see, they really like, the families really like they enjoy the process of being assessed, because it's a very nice way to understand their infant's development, and we also give them feedback. So we have this, we have Instagram, we have a website. Maybe you can, if you are interested, you can look later. So a lot of the families like to post some photos and stories on Instagram to show how much their babies like the project. So this is the broad domains that we have in the Germina cohort. So of course we have the brain measures, and brain measures, I mean the EEG that we have in the project. We collect data about the environment, and here I use the word environment very, very broadly. So here I'm considering maternal mental health, I'm considering the home environment, risk factors, violence, and stuff like that. We also have behavior and cognition. We have a lot of scales trying to measure early signs of behavioral problems, mental disorders. We have the developmental milestones using the Bailey, social communication using the Vineland, and so on and so on. It's so many measures, it's not possible to mention all of them now. We also have, we collect biological data, like I mentioned before, genetics and epigenetics. So we have saliva from all the children. So we have their genetic profile and also their epigenetics profile, and also the gut microbiome. So we collect feces at 3 time points specifically to understand the role of the microbiome in influencing behavioral, cognition, the brain. And it's a very, very hot topic these days in health sciences. And our idea is to grab all these domain, all these data, all these variables, and implement some data integration models, because we collect so much data these days that it is not possible to run like the good old t-test, the good old linear regression. It's simply not feasible. We have like literally at this point thousands and thousands and thousands of variables. If you consider only the gut microbiome, for instance, Grover here probably knows better than most, we have thousands of variables. So it's not feasible to do like the good old statistics. So we have to implement some complex models to deal with so much data to try to predict executive functions, developmental milestones, and mental disorders, and so on and so on. So for the purpose of this presentation, I will show some of the preliminary data that we have at this point. We just submitted our first paper from our cohort. It's more of a methods paper, in fact. Our cohort, we have so many variables, so much stuff that we thought that it would be very important to have a methods paper, a paper that we could keep referencing in the years to come to help reviewers and our audience to understand our cohort, so we don't have to explain every time again how we did all the data collection. And it's not only a methods paper, but we also included some of the preliminary results considering only baseline, so only the data from 3 months of age, okay? It's probably a little bit difficult to see, but this diagram here is showing our recruitment process. So we did mostly online, social media, and mouth-to-mouth recruitment. So we started with more than 1,000 potential subjects, and then we selected them based on some of the inclusion criteria that I can mention later, or you can read on our paper. So we ended up with 557 mother-infant diets. Okay, so this is our final sample at baseline. And this is a little bit of the characteristics of our sample. Okay, so this is the basic social demographic and economical characteristics at baseline. So since our recruitment was like I mentioned before, based on social media and using websites and Instagram and Facebook ads, we have a little bit of a bias. We don't have a representative sample. This is very important to understand. So we have a bias in terms of I think the most important variable to mention is income, family income. So we have families that are mostly middle class and upper middle class. And also in terms of skin color, the majority of them are white. So considering that Brazil, it's a very diverse country, we don't have a representative sample. And yeah, so I don't think it's worth mentioning every bit of information here, but I think the takeaway message, it's not a really representative sample, but it's a very rich sample in terms of data, like I mentioned before. We have a lot of important data. So for the purpose of these findings, what we did is to take the Bailey scales. I believe most of you are probably familiar, but if you have some questions, we can answer later. So it's like the gold standard to assess development milestones these days, even though it has its flaws. So what we did is take all the 5 raw scores that we have in the Bailey. So cognition, language, and motricity, and we took all the raw scores and used a CFA model, a confirmatory factor analysis model, to extract a latent variable. Okay, so this latent variable composed of all the 5 raw scores was our outcome for the purpose of this analysis here. So it's basically a variable indicating how well the infant is developing at 3 months of age. And then we considered all these predictors that you see here. So these are broad domains, okay? So we considered socioeconomic and demographic variables. We considered maternal mental health, home environment, infant behavior, anthropometry, genetics, epigenetics, EEG, and microbiome. So inside each of these domains, we have dozens of variables, in some cases hundreds of variables, and in some cases thousands of variables. Okay, so it's difficult to see, but it's just to understand a little bit the number of variables that we have in each domain. Okay, so in our paper, you can send me an email later if you want to read. I can send you the link, and you have more information. But this is basically all the variables that we have across all domains. Okay. So what we did, thanks to Grover, who is here in the audience, is to implement a machine learning model to try to select these variables. Okay, so this is a little bit technical and complex. If you have more questions, maybe we can answer later. But basically, the idea here is to find the best features that can predict our outcome using this idea of optimizing your data. So this is basically data-driven sparse partial least squares model that we end up, we can all the way to the data and to find the best features to predict our outcome, okay? So across all the domains and variables that I showed, we have these results that you see here, okay? So this is a coefficient plot. In the y-axis, you have the coefficient of a linear regression. And here in the y-axis, each row is a domain that I mentioned before. And each of these dots represents the coefficient. So the domains that showed that had a significant relationship with the outcome were socioeconomic and demographic variables, two EEG variables, one with a theta band and other with a beta band, epigenetics, and microbiome. All the other domains, we didn't reach a significant p-value, okay? So this is our main results at 3 months of age. So as you can see, we collected so much data, a lot of data, all the trouble, all the money spent, and then we end up having just these domains showing a significant prediction of our outcome, at least at 3 months of age. We don't know much about the other time points. And here, this second plot, it shows the what we call the relative importance. So each domain that I showed contributes to understanding the outcome, right? So here we have a percentage of how much contribution to the model. So if we remove each variable, how much would we lose in terms of information? So as you can see, the socioeconomic and demographic domain had the most importance. So we spent millions collecting biological data, and then we used some like very simple questionnaires asking some questions, and we have the best predicted value here. But of course, also the brain measures, specifically the EEG, was also very important. So yeah, and as you go down here, you see that some of the variables are not that important, okay? So I think that's it for now. I just wanted to describe very briefly our cohort, just to show what we have been doing together with Rogério and Vicky, and also show some of the preliminary findings that we have. So one very important message for today, I would say, is that our cohort it's not publicly open, but we are open for proposals. So if you are a master's student, a PhD student, you can contact us, and we have a website with our code book. So we have like a data dictionary. We have all the variables, so you can look at our variables and you can propose an analysis, for instance. So I'm very much interested in the role of parent-child interaction in cognitive development using the Bailey. So you can actually write a proposal for us, and then we assess the proposal, and then we can give the data. So we just have to make sure there's no overlap between the postdocs and students that we have working on the data now, no overlap between papers, and so on and so on. But we are very much open to people working with our data, because as you saw, we have thousands of variables, but not enough people to analyze. So we would like to have people to work with us, working with this data. And of course, I was not able to show everything that we're doing with this cohort, but you would be able to ask questions later. And then you can also email me, feel free to email me if you want. And you can also read our paper, the methods paper that I mentioned. It's not published yet, but you have the preprint, so you can actually read everything that I showed and understand all the variables. There's much more than I mentioned here at this presentation. So thank you for your time, and yeah, so that's it.
Thank you, Daniel. So would you like to stay here or go to the seat? while Vicky presents her
Yeah,
work?
I think I can stay there just to see a little bit better her presentation, and then maybe you can come
Okay,
here
okay.
to discuss
Yeah.
later. Yeah.
Oh, absolutely. So I would like to mention that one of the analyses that we'll begin to do in this big project, Germina, is the assessment of daycare educational units. And we must begin this in August. And 2 students of this assessment research are here. Isabella is a PhD student, and Caroline is a master student who probably will have her research turned to a PhD one in the next evaluation of her dissertation. So we're very happy that we could form this group of researchers, and we will contribute in the future to assess some environmental variables to this very large study, as you have seen. So, Professor Victoria Leon, please, the mic is yours.
So thank you so much to Rogerio and to Lizzie for the invitation to speak with you all today, and also to Daniel for the fantastic introduction. Indeed, it's been my absolute pleasure to develop this collaboration with the wonderful colleagues here in Sao Paulo. This is actually only my second visit to your beautiful country, and both times I've been impressed by its rich cultural heritage and diversity. And I think that's one of the things which really inspires me as a developmental psychologist, looking at the context in which brain development, child development happens, and how the environment shapes each child's individual journey through life. So, as introduced, my name is Victoria. You can call me Vicky. I have multiple hats. I won't go through all my different titles, but suffice to say that I live and work in 2 countries. Most of my time I'm in Singapore running a research center, the Empower Research Center, but I also am in Cambridge University every quarter to lead my research group, which is also based there. So I have 2 bases from which I can conduct research. Now, today's talk is about parent-child interactions and relationships, and it's only one slice of that wonderful cross-sectional view of the multiple factors that shape child development. But it's a really important one because the parent-child context is one of the most proximal environments that incubates the child's brain and cognitive development. It's one of the strongest influences that the child will be exposed to and has a disproportionate effect during early life on the lifelong trajectory. So what Daniel showed in terms of the demographics and the socioeconomic status influencing Bailey scores, a big proportion of that is going to be effects through the parent-child interaction and home environment. So my work is trying to understand these mechanisms. How do these differences in parent-child interaction embed themselves in the child's brain, under the child's skin, and shape different trajectories? So let me start by painting the bigger picture. I'm sure many of you who study psychology don't need to be convinced about the importance of the early social environment. But whenever I talk to government officials and economists, increasingly I have to find the right language to make the case for how important our field is. And when I do this, I like to appeal to a review that was done in Nature nearly 15 years ago now, by a group of scientists commissioned by the UK government. This was the Foresight report, and the government was trying to find out, projecting into the next few decades of life, what kinds of human skills should we focus our energies in researching and developing to make sure our citizens are going to be best equipped to deal with challenges? And these group of very clever scientists came up with a way of describing the key psychological skills and abilities that we know are important, but convincing the decision makers about why this is important. And they used the term mental capital. This is probably the first time that appeared in print. It was deliberately chosen, the word capital was chosen to give the idea of like a seed fund or an investment. There's a store of potential or capital that a child develops over life. And you start with a certain amount, but if you invest the right way, that amount can grow. And I think that was very smart because, it puts into concrete terms what we as developmental psychologists know, that children's trajectory is malleable. And the environment and what governments do has a really important role in developing that capital, or indeed, it not actually going to plan. But also importantly, they highlighted that there were two broad domains in which we need to think about capital. There's cognitive capital, intelligence, memory, executive function, all very important skills. But there's also emotional capital. And 15 years ago, this was not well recognized. People didn't see the equal importance of social emotional skills as being part of your store of capital that you need to develop as an individual to be well-rounded. So I think this was really smart of that group of UK researchers at that time to phrase things this way. And I still like this formulation now. So what types of skills are we interested in? Well, in terms of what can change over time, some of these cognitive and emotional skills are more malleable than other skills. So I gave the example of intelligence. And because intelligence is so highly heritable, there's less we can do to change it, unfortunately. If you have severe deprivation, or if you have some kind of environmental insult, then probably you're going to be mentally impaired in some way. But otherwise, it's very hard to improve your intelligence, despite what all the games and apps will tell you, or what baby toys will want to claim. It's really hard to change intelligence. But there are other skills, and in particular, some of the executive function skills, like mental flexibility, which is one of the EFs, has higher environmental contribution, which means your experiences, both in the home and in the school and in the preschool, have an impact on changing your amount of flexibility, your ability to allocate your attention, your strategies, and all your cognitive resources in a goal-directed way. So we have to be smart as well when we think about where we want to put our energies, because some skills are more malleable than others. So flexibility and social resilience are two which are more malleable compared to other types of cognitive constructs. And of course, it then, relates to this, that the best time to try and improve this capital is early in the child's life, during the sensitive windows, where the brain circuits are still open, plastic, and malleable, and where the environmental input has disproportionately high impact on later development. So that can be illustrated in this curve. Ooh, sorry, it's so tiny. On the y-axis is a measure of potential or brain function. On the x-axis is developmental stage, so birth, early childhood, adolescence, and adulthood. And this curve shows this change in the child's potential or developmental status. And as you can see, the change is rapid over the birth and early childhood years, and then starts to kind of stabilize more over adolescence and adulthood. And what this shows is just that difference in the starting point of a child comes from the balance of risk and protective factors. That's the early life adversity model here. But also, it's not deterministic, because if you come in at an early stage with intervention, you can change or uplift the child's trajectory. And so you can increase or make more optimal the child's developmental potential by giving the right balance of intervention at the right stage. Now, this is a nice picture, and I think as psychologists, we broadly accept the limits of this proposal. The challenge is, in practice, it's really hard to implement a model like this. So when I talk to the government, they'll say, yeah, okay, you've made the case. Early childhood is important. We want to do stuff that helps. But exactly what do we do? What is the target? How do we know what to give to each child? And that is the problem with measurement, because you have this putative scale here of risk and resilience, but we don't have good tools to tell us where does any one child lie on that scale, and therefore what should they receive by means of intervention. So this is the problem now with translation, and it's one of the reasons why in LEAP I was asked to develop measures and tools. It sounds really boring, but when it comes to translating theory into practice, the tools are really important. And our current tools are really limited in how specific, how precise, and how early we can use them to identify risk and therefore to give good interventions. So that's where my work has come into play. And just to also say that I'm really interested in diversity, and that's one of the reasons why I have this 24-hour commute between Singapore and Cambridge, and I maintain 2 research groups, because I think it's really important to understand the origins, developmental origins of natural diversity, how we grew up in different contexts, how our brains change in a way to allow us to thrive in different cultural countries, linguistics, and all kinds of ethnographical differences allow us to thrive in those different contexts. So it's very interesting for me to figure out what is common across different cultures and seems to be quite universal, and what is really driven more specifically by the practices and what's reinforced in your specific culture. So that's one of the reasons why I bother to do this in 2 countries. Okay, so now specifically, what do I measure? So one of the things I'm really interested in is the parent-child as a dyad. So those of you who study development will know that very, very early in life, the child and the parent have a really closely attuned relationship. If you watch them, you can see that they share a lot of their states together. If the baby's unhappy, it's very hard for the mom to remain stone-faced and to stay happy. She's going to respond to that too. And vice versa, if the mom's in a very positive, healthy state, her baby will catch that emotion and be contagious to that emotion too. So these two, the mother and the child early in life, are very permeable ecosystems. And in fact, some people, like Daniel Stern, have described their interactions as a dyadic dance. Almost like their behavior is like a bit of a tango. Give and take, serve and return, push and pull. There are very strong connections between them. And before I started looking at this field, others have already described this in terms of biobehavioral synchrony. That's coordination in behaviors, but also in your body and your physiological changes and your responses. When I came into this, I thought this was really fascinating. And I thought, if we see such behavioral coordination, it's natural that the brains must also be coordinated in some way to permit that coordination to occur. And so I started studying synchrony at the neurological level. And one of the things I realized very quickly on is that it's quite a transient state. So these synchrony states between the mother and the child, it's not something that is maintained for very prolonged periods necessarily. These are transient states. You couple and decouple, couple and decouple. So these are states that you go in and out of. And so it became apparent to me that much as I'm interested in synchrony, it's perhaps even more important to understand what drives synchrony. How do we bring people together for a few minutes or a few moments? And then while you have that golden period of temporal coupling, what changes in the brain? What is easier to do and harder to do? So I became interested in these dynamics of synchrony. So I started to study the context in which you get naturally increased synchrony. And these are when you start to use cues, like we've studied eye contact or gaze, certain types of emotional vocalizations and facial expressions, etc., which seem to have an effect in boosting the capacity of 2 people to couple together. And then, like I said, we're interested also in why we do this. Why bother to synchronize with someone else? What does it create in terms of the mental states? So other people, like Ruth Feldman and other colleagues, have studied affiliation and bonding, and certainly this is one very important outcome. If you have consistent, healthy coupling, you tend to also form longer-term, more stable bonds. But because I'm a cognitive psychologist as well, developmental cognitive neuroscientist, I like to study the impact of synchrony on things like how well we communicate and understand each other, how well you're going to learn from me and I'm going to learn from you, and of course, how that changes our decision-making about whether we explore the world, whether we take risks, and how stable our cognitive states are. So I studied the more cognitive aspects of synchrony. And how we do this is with a technique called dyadic EEG. So some of you may be familiar with normal electroencephalography, where one person wears a cap and you usually watch something on a screen, and we record brain signals that are elicited in response to one stimulus. In dyadic EEG, it's much more complicated. We collect brain signals from two people, and they are at the same time responding and stimulating each other. So it's a much more complex dynamic than one person watching, one directional input, right? You have this dyadic interplay of I'm stimulating you, but I'm also reading your signals. And it's that kind of circular relationship, recursive relationship, which we have to be able to model somehow. So we do that using techniques, modeling techniques, so you can't use simple averaging for this one. We use modeling techniques like coherence, a Granger causal measure of that, like partial directed coherence, and then we can pick apart influences of the mother on the child and vice versa. This is not the only one. I'll show you other techniques that we also use, but this is one of the first few that we started working on to capture these bidirectional influences. Okay, so I talked about the social context in which we generate synchrony, and as a psychologist, I gravitated naturally to this idea of ostensive signals and natural pedagogy, because it has been proposed that the human species over time has fine-tuned this range of signals, which enables us to very quickly and very reliably highlight information to young members of our species that tells them the communicative intent and the relevance of the message. So when you're a young baby, there's a lot going on in your world. Lots of sights, lots of sounds, lots of people, lots of stimulation. And one of the things that caregivers need to do is focus your attention on what's relevant for you to learn now. So the idea is that natural pedagogy makes use of these signals to allow you to very quickly signpost to a child, learn this now. And these signals are things like eye contact. I'm looking at you, so pay attention to me, all right? Using the child's name. Hey, Rogero, look at me. And infant-directed speech. So when we talk to babies, we don't speak like me now talking to you. We have this much more melodically varied tone, and we slow down, and it's colorful. Wow, that's much more interesting. That's called infant-directed speech. So the color is very different, and all of these are types of ostensive signals. They're perceptually really salient. The baby gravitates towards them and kind of intuitively understands that when they hear or they see these things, this message is for them. And also, those of you interested in psychiatry will know that if your mother has a disorder, a mood disorder like depression particularly, the use of these signals becomes much more impoverished. They're not as contingent, their speech is much more flattened, they don't color as much, they don't look at the child as much. So there's much less effective use of this signaling, social signaling system, which means the connection between the mother and child breaks down a little bit. And we know that has an impact on developing cognition, particularly developing language. Specifically. So I started by looking at this system because I had a suspicion that this system may activate the synchrony that I'm so interested in measuring. So my early career, and I won't go into much detail because there's a lot of technicalities here, was looking at child-directed speech, that melodic register. Now, when you hear child-directed speech, most of you will be drawn to the pitch changes and the frequency changes. But what I showed in this body of work is that there's very nice changes in the rhythm and the temporal structure of the way that we speak to children. It's much more regular, and it's much more predictable. And in fact, when you decompose the temporal structure of child-directed speech, you get these characteristic peaks in the rhythm signature. And they come out at the level of the stressed patterns, the syllable patterns, and the onset rhyme patterns. This is English, I should say. Portuguese is not a stress time language, so yours is going to be different. In English, which is stress time, we get these three key peaks in the rhythmic patterns. And to cut a long story short, what was really nice is that these rhythmic signatures match the EEG rates that are endogenously there in our brain networks. So Daniel highlighted one of the predictors is EEG theta. The theta rate matches beautifully with the syllable rate of child-directed utterances. So it's almost like when we speak to children, we emphasize the temporal rates that their brains are prepared to receive. They have these oscillations which are already tuned to the natural rate at which we would produce syllables when we talk to children. So there's this privileged match between the delta stress rates, the syllable theta rates, and the alpha onset rhyme rates, which suggested that there's a speaker-listener match or speaker-listener entrainment that could help the child break into the speech signal. So that's what this work showed. So that was my first foray into looking at synchrony, but from this perspective of rhythmic signals and the speaker and training the child's brain with rhythmic signals like child-directed speech. So my next piece of work built on this, and I said, all right, I think speech is going to have one of these abilities to help us synchronize and couple with children. What else would work? So this is probably the most well-known paper that came out of my team. And we looked at whether eye contact also was good for increasing synchrony with children. And in this experiment, we had the adult here looking at the child straight in the eye or looking away from the child, so indirect gaze. But in both cases, she was singing a familiar nursery rhyme, like the wheels on the bus go round and round. So there was this nice regular entrainment context from the speech. But on top of that, we either had direct gaze or indirect gaze. And we were curious whether the gaze would also modulate the amount of synchrony between the adult and the child. And as predicted, we did find that. So using this directional measure, we could show that both in the direction of the adult to the child, as well as from the child to the adult, the synchrony was higher when they shared eye contact than when there wasn't any eye contact between them. So this was a bidirectional effect which showed the power of mutual eye contact in raising brain-to-brain synchrony. Now, it wasn't just the eye contact in this experiment. We also could look at the effect of the adult calling the child's name, because sometimes the child was inattentive. So the adult called the child's name to get them to orientate back to them. And we could show that before and after the adult uses the child's name, there was an increase in synchrony after the use of the child's name in both theta and alpha frequency bands. So this multimodal effect seemed to hold in this communicative context for both eye contact and using the child's name. And then finally, just to close that loop, because these were fun nursery rhymes, which were familiar for the kids would babble along or coo along. And so my research assistants hand-coded all the utterances from the babies, and they scored how many vocalizations and how long the vocalizations were. And so this plot shows you on this y-axis scale there, each child's average vocalization duration, and each dot is one child. And we showed that the more the child vocalized, the stronger the effect they had on synchronizing the adult. So basically, this little experiment was showing that in this small microcosm that we set up, we had multiple synchronizing cues. We had the adult looking at the child to increase synchronization, calling the child's name, that also increased synchronization. But then that seemed to release the child's vocalizations. And the more they vocalized, the more they reinforced the synchrony within that dyad. So we got this positive feedback cycle driven by the exchange of these multimodal social signals. And as expected, a lot of these signals are the ostensive cues that I told you about earlier. So we think that this probably tells us there are effects that these ostensive signals have on generating and sustaining synchrony between listener and speaker dyads. In another study, we also looked at emotion. So this is a study with positive and negative affect. Mums were showing pleasure and delight and approval, or disgust, negative, disapproval faces. And we measured the brain-to-brain coupling of the mother and the child in these two different emotional states. And to cut a long story short, when we applied these graph network indices, which we developed specifically for cross-brain measurement, we showed that positive emotion makes for the merging of the mother and the child's brain into one single hyperconnected brain. You can describe it better as one unit than as two separate units. But when the emotions are negative, this hyperconnected brain breaks down into two subunits of the mother and the child. So positive emotion helps to integrate the two mother and child networks into one superconnected network, which is quite a nice finding here because it suggests that information transfer is going to be much more efficient with the positive emotions, and also may give us a nod to why in depression, if the mother is always a bit negative, those two networks always segregate. So it's very hard for them to couple and exchange information. And so the parent will have less of an effect on driving the child's development. And for those of you who are interested in the methodology, we applied two different measures here and we found consistent effects, whether it was a directed measure like the coherence or a non-directed measure like the phase locking value. Okay, so that I hope gives you a flavor for my work and why we wanted to use this, psychology background to study how it drives and creates states of synchrony. But I now want to move forward into what we can do with this. And that is one of the reasons why we joined LEAP and why we started working with Daniel and Guilherme, which is to try and develop predictive models based on all these really important social signals that parents exchange with their children. So this is a technique that we call dyadic neurosociometrics. And so we know all these signals are important in generating a state of coupling between the parent and the child. So in this technique, we concurrently measure multimodal signals in gaze, in posture, in EEG, ECG, speech, and emotion concurrently from the adult and the child. And we compute temporal contingencies between them. And we generate nearly 1,000 indices. So Daniel mentioned how many thousand. We on our own have about 1,000 indices coming out of this method per 10-minute interaction already. And we use that in computational machine learning models to identify profiles or fingerprints that we can then see if they map to different pathways or trajectories of development, particularly if they're developmental disorders that have different fingerprints. And one of the things we were doing in LEAP is trying to speed up this process because a lot of these indices have to be hand scored by research assistants, what was the baby's emotional state. So a lot of our work is now in automating that using machine learning models. And so here is one example of our body pose model. And you don't even need to wear a motion capture suit. You just need the videos now. And the machine learning does all the heavy lifting. It guesses where the key points of the child and the mother are, and it spits out information, XYZ information at every single frame for you. So that really cuts down the amount of hand annotation time that you need to be able to get very quickly these measures of coordination and synchrony between the parent and the child. Okay, so just a quick glance now moving forward where we're going with that. So the other projects in my lab are now looking at developing that sociometric profile, the predictive profile, into a clinical tool to help GPs and clinicians make more informed decisions about triage and referral onwards. And we try and give them very detailed information, but very accessible about these domains and how the child is performing across them, a traffic light system for where the child is doing well and maybe not so well. And importantly, because we can do this so young. We can do this at 3 to 6 months of age, right? It's much earlier and much more accessible and scalable than current screening tools. But we're also conscious that at this really young age, it's maybe not helpful to give a child a hard label. You don't want to say that a 6-month-old is autistic. It's in some countries, like in Singapore, that just is not really helpful at that young age. So we want to maintain this transdiagnostic approach where we look much more at the shared symptomatology and the key domain performance information with the main aim here of guiding intervention. And that brings me to the second part. I'm afraid I don't have time to talk a lot about this, but we've developed toys that are smart that can take these profiles and give live information back to the adult about the child's cognitive development and performance. So we're also developing play-based interventions that are based on these sociometric profiles that we computed. Okay, I'm halfway through my talk now, and I hope I've taken you through the psychology of it and into the translation. But in the last part, I just want to satisfy some curiosity, because whenever I give these talks in a scientific context, they all go, yeah, this is great, but how does it work? How do we get synchrony in the first place? So there's a big part of my research, which is figuring out these mechanisms. How do we generate synchrony in the first place? So if you bear with me, this is going to be a bit more technical. I'm going to talk about adult and animal models. So we're going to go out of babies, but it will help us to answer a bit more of this question of how do we generate these synchrony states in the first place? And are they really causally effective in generating the behavior that I've shown you? Okay, so first of all, a little rant, because every time I talk about synchrony, people come away with very limited views in terms of what I'm talking about. So when I say synchrony, most people think about, people marching in step. Left, right, left, right. I go up, you go up. That's really not what I'm talking about here. Synchrony can occur in many different states, and we're talking about contingencies that can either be linear or non-linear. Most of the way that we understand synchrony is linear, which means we talk about these entrainments, right? So up and down, up and down. And yes, that's one way that you can see synchrony, and you can measure it using things like the phase locking index, GPDC, etc. And it's a very simple, I go up, you go up, or I go up, you go down type of relationship. But in real life, especially with human behavior, it's quite rare to get that really restricted type of relationship. You know, when we talk about synchrony, maybe you want to think about a couple doing a tango, right? And you know when they have good synchrony, when they're a good pair of dancers because they're really well coordinated. But it's not the case that the man and the woman are doing the same thing at the same time. In fact, you never, very rarely get that unless they high five. Right? It's most of the cases that I step back, you step forward, I do this, you do that, and it may not even be the same time. I start with one, and then a little bit later, you do something else. But there's very good coordination between the two of them, and there's the same overarching temporal time frame which they are obeying together. That's a very complicated relationship, non-linear relationship. So to capture that, and I think that's what we see much more often in human behavior, we need non-linear measures. So increasingly, we're developing other techniques based on information theory, which looks at distributions and probabilities, and dynamical systems theory, which looks at states, trajectories, and attractors. I'll give you one example of this later, to capture these non-linear dependencies. All right, I'm going to stop here. This is just my rant, because people always seem to have this very limited view of what I'm talking about. Okay, so a little bit more in terms of techniques. Those of you who are interested will know that there's a lot of intricacies when we select the right variables. So we did one exercise to help us determine what was the best window length to use. And we found that, you know, depending on whether you were using a linear or nonlinear measure, looking at the child's brain or the adult's brain, there was this double dissociation just in this one parameter on the best window length to use based on an intermediary criterion like signal to noise ratio. And we found, interestingly, that this was related to a developmental switch in the preponderance of linear and nonlinear coupling in the two brains. So when you're a very little child, a lot of your connectivity can be well described by linear oscillations and communications between different parts of the child's brain. But as you get older, and in particular the adult brain has a lower linear connectivity and relatively higher nonlinear connectivity. So this is one of the reasons that explains why you can't use the same setting for a child and an adult analysis, because development has shaped how the brain communicates. So it's not one size fits all. So this is quite a nice point, saying why we need nonlinear measures, because if you want to capture everything, you have to have both sets of tools in your wheelhouse. And then this last bit is to just illustrate what I mean by dynamical systems theory. This is simulated data, actually. And what we do here is we try and map the landscape of possible cognitive or energetic states that any one or dyad of animals could occupy. So we don't assume that you're either in or out of synchrony. There's a landscape, or we call it a manifold of possibilities. And you navigate that landscape based on the current cues, your background, your history, your neurochemical current state, and that shifts you in different part of that landscape. Now, there's certain parts of that landscape which you're more likely to occupy, and these are illustrated with these troughs here, these deep wells, which we call attractors. Attractors are parts of that landscape which you're very likely to sit in because they're very attractive. And if you think about children who are very perseverative, very recursive, they have very deep wells there where they keep repeating behavior because that's a very attractive state. And part of development is trying to get them out of that into a more flexible way of thinking or behaving, right? And the parent has a very important role to play. But this is one way that we can model in a much more sophisticated way the realm of possibilities of cognitive states that a child, with the parent's input, can occupy. So these are this is me trying to bring you through an evolution in more sophisticated ways of understanding and capturing, the states which the social partner can help you to occupy. Okay, so coming back to entrainment. Does this happen? Yes. So there are linear effects in the brain already, and this was published back in 2022 with my colleagues at Cambridge University. And we showed that there were really interesting effects when you can entrain the visual cortex on effects on perceptual learning. So in this experiment, the task was really simple. It's perceptual discrimination. You were shown either this pattern or that pattern, and you had to say if it's concentric or radial. Very simple target detection. But before the target, we flickered some visual boxes at a rate that would entrain the visual cortex of the viewer. And these, I should mention, are undergraduate psychology students at Cambridge University. So they're very bright, no IQ difficulties there. But the trick here is that we designed the flicker to precisely match the rate of their alpha oscillations. So if you're a neuroscientist, you know alpha is the most dominant rate of oscillation. You can see once your eyes are closed. But all of us have a peak rate. Everybody falls roughly within 8 to 12, but every individual has a particular rate that they oscillate at. So we measured that rate. Let's say for one person, it's 8.1. Another person is 11.5. We measured the ideal peak rate, and we delivered the flicker exactly on their rate. And so we had this matched flicker, and in another group, we did a non-match, which means if you're 8 hertz, we give you plus 1 hertz, too fast. Or if you're 11.5 hertz, we do 10.5 hertz, too slow. Okay, so we have either matched or non-matched flicker rates that would entrain the visual cortex. And then we delivered this target either at the peak of the entrained oscillation or at the trough of the entrained oscillation. So it's a 2-by-2 manipulation, rate by phase. And to cut a long story short, you can read the paper, we got these really dramatic effects on learning. Like I said, these are Cambridge undergraduates, so they have no problems learning. But you can see here that in the condition where we gave them the matched entrainment rate and we delivered the stimulus at the trough, they learned 3 times as fast, 3 times faster than a control group with no flicker. So we were boosting perceptual learning by targeting the entrainment, and then target comes in right at the trough. By contrast, when we gave the wrong rate, too fast, too slow, or we put the stimulus at the peak instead of the trough, these two groups flat. They showed 0 learning across nearly 3 hours of practice, 900 trials of practicing this. They didn't improve at all. Like I said, no problems with intelligence, no problems with paying attention. They were paid. But basically, the information was not hitting the networks at the right rate, and the information wasn't getting passed on because it wasn't matching the endogenous rates of coupling. It comes back to my idea about infant-directed speech and why we have this match between the speaker utterances and the rate of the child's oscillation. So when you mismatch things, you really break down the capture of information. This is what this is showing in a very basic visual paradigm. So we know these basic gating effects can happen. We can entrain oscillations and either drive learning up or flatten it completely, just at the basic information capture and encoding level. So that's quite fun. Of course, we never do this with babies. We can only do this with adults. And so we're now thinking about developing this into BCI technology, because each of us has a different rate. And so you can think of a way to then figure out what your ideal rate is and then train stimulus delivery so we always match your preferred rate. And that could work potentially for some children and children with maybe atypical developmental trajectories whose oscillations also have a different signature. So this could also be helpful for them. But I now want to come to much more invasive work, because the only way to really strongly test whether synchrony matters is to be able to dissolve it or force it at will. And it's really hard to do that in a human model without being unethical. So I have to work with rodents. So before we even started this, we did our due diligence. We tried to understand rodent development, mouse development, and we tried to map similarities between the human and the mouse developmental stages. Luckily, we go through similar stages of development as mammals. We do still have infancy and adolescence as a little rodent pup. And we also tried to see whether there were analogies in the social behavior and how that emerges. Now, there were big differences, first of all, in the parent-child relationship, if you can call it that. First of all, rodent dams, mummies, mouse mummies, are terrible parents. This is no surprise because they have not evolved any further. And I think part of it is because how terrible the parenting is. You know, they really ignore their children, they abuse their pups, and worse come to worse, they eat their pups, and they're completely unresponsive. There's no bidirectionality naturally, okay? It's everyone for themselves. So these dams really don't go out of the way to care for their pups at all, right? Of course, in humans, it's very, very different. We do have much more bidirectional contingency, sensitivity, etc. There's a very big difference in the rodent-dam-pup relationship. apart from that very big difference, a lot of the basic kind of licking, grooming, feeding, nutritional, peer-to-peer interactions seem to have analogies across humans and animals. So with that one big caveat in terms of parenting quality, a lot of the other emergence of social behaviors seem to be underpinned by similar capacities across the species. So if you're interested in that, you can read our review article, which was published last year. But that then gives us confidence to know what to target and what to expect out of the rodent dyads. So this is now hot off the press because this is about to go in, but we created optogenetic mouse models. And we created a model of a mouse mummy and a mouse baby that you could synchronize at will and desynchronize at will. And you do this using optogenetics. So optogenetics is a way of splicing a light-sensitive gene into any neuron or cell type that you care about. So we chose the prefrontal cortex, the parvalbumin GABAergic interneurons. So we spliced this gene into the mouse medial prefrontal cortex and into neuronal populations that are responsible for gamma circuitry. And we could do this in the dam and in the pup so that when you flick on a light, you activate the neurons and you force them to synchronize. Similarly, if you trigger the light signals at random patterns, you force them to desynchronize. So we could, at will, with a light switch, create perfect synchrony or create perfect desynchrony. So we had a highly manipulable model that we could then use to test our causal hypothesis. So, all the questions about, is synchrony real? Does it really matter? We could test the downstream consequences with this particular model. And so we started off, this was our first experiment, with a paradigm called social transmission of food preference. This is analogous to social learning in children. And so social learning is where you learn vicariously. You don't experience it yourself. You watch somebody else or you interact socially and you gain knowledge secondhand. This happens in mice too, and the analogy here is with learning about food and food safety. So the idea is that one of the mouse, in this case the mummy mouse, will be fed a novel food, just like they would when they go out to forage in a novel forest, eat some new food. And they come back to the nest. And then they will interact with their peers or with their pups. And through that interaction, the pup will smell and get information about this new food that they then learn vicariously about, without having eaten it themselves. And then later, when you take the pup and you give them a choice of two foods, either the food that the mummy ate or a different food, they will eat the food that the mummy ate more. That's because they learned vicariously that the food the mummy ate is safe to eat. So they learned it secondhand without having gone out to explore themselves or eaten themselves. So this is vicarious knowledge transmission, and similar to social learning in human babies, which they can do by about 9 to 12 months. So we did that food preference paradigm, but we did it in the context of the optogenetics. So the first phase where the dam went off and ate the novel food was the same, but during the interaction, we either synchronize or desynchronize the two brains. And this was because we wanted to see whether the synchrony during the interaction was important for information transfer or not. So during the time when the dam came back and was interacting with her pup, we either forced them into a state of high synchrony or we desynchronized them. And then afterwards, we isolated the pup and tested them on whether they would eat, which food they would eat. Okay, so this is the paradigm. And we also did it at this P21 stage, which is late adolescence in the pup's life. So here is our main result. I'm pleased to say that we did have an effect of the synchrony. And the effect was a really interesting one. It was not appetitive behavior. We didn't change how much food the pups ate per se. That was more predicted by their weight and how hungry they were. What we changed was their social foraging behavior, because this raster plot shows you how much time they spent in the demonstrated versus non-demonstrated bowl. The synchronized mice, even after they were full, so this is the part when they were eating and relatively hungry, by now they're pretty full. Even after they were full, they were still hanging around the bowl with the mummy's demonstrated food scent. So it tells you that it's not about the hunger, it's about something of the social value of that scent or that stimulus, which is still lingering and still driving their behavior. So it's socially motivated foraging, which we seem to change in this particular experiment, which is quite good because we were stimulating prefrontal cortex, and that's related to reward and reward learning. So it's not so much about how hungry you are. But then, here's dynamical systems theory. We wanted to go further because we were not happy with just this very simple description. So what we did was we then applied some kind of state space modeling to look at how the sync and desync groups were behaving differently, the range of behaviors that they could exhibit. And without going into too much detail, I'm very happy to answer this later. We modeled the Helmholtz free energy of the system, the temperature of the system, and the entropy of the system, which would give us a 4D dynamical systems equation of state for the state space. Long story short, we showed that the synchronized pups had systems which were much more stable. That's why their state space is lower on this energy plane and had generally lower entropy in terms of the foraging behavior, which means whatever they learned had created a much more stable state, a range of behaviors that they then subsequently showed. Okay, we also looked at how in the moment social interaction, because remember we simulated them during social interaction, we looked at how the social interaction itself changed. And here my poor students, research assistants, hand-coded the animal behavior. They were like, Prof, why are we coding rats? Sorry. So they were looking at frame by frame at how these mice, mummies, and babies were, playing with each other, and they were coding all kinds of things. So, normal things like close proximity, approach behaviors, social investigation behaviors, but we even looked at how often they were sniffing different parts of the body, the head, the abdomen, the anus, the tail. We coded 19 different behaviors, and you could see across nearly all of them, and the effect sizes are here, nearly all of them, when you synchronize the dam and pup, these behaviors are much more prolonged compared to when you desynchronize them. So there was an in-the-moment acute effect of prolonging and sustaining social interactive behaviors to do with approach and investigation. And then we looked at this over time as well. So that was the average, but this is over time. So the interaction was about 15 minutes. We saw this really interesting pattern where overall, the synchronized diet spent much more time just in close proximity to each other. And this is significant because at this pre-adolescent stage, the mummies are trying to distance themselves from the adolescent pups. They're trying to get them to wean and go away. Like I told you, they're terrible parents. And so they want them to be gone. So naturally, they spend very little time together. But in the synchronized group, they spend much more time in close proximity. And what we noted was that at around the 10-minute mark, when we looked at what qualitative behaviors were happening, the synchronized families were showing nesting behavior. Really unusual. The dam was settling down in a corner, and her babies were coming to her and huddling together. And we only saw that in the synchronized groups. We never saw that in the desynchronized groups. In the desynchronized groups, both the mummy and the pup were just running around doing their own thing, right? So we had clearly changed not just in the moment dynamics, but even the kind of parenting, nesting behavior that would kick in much later and potentially driven by the oxytocin system. So this is quite nice. We could also capture the recurrence of this type of behavior. And again, we show, so this was RQA measures, higher temporal contingency and higher stability for these behaviors. So to cut a long story short, we can then put those two things together. We can model how the social behaviors are then creating this much more stable form of exploration that we see in the pups later. And we could describe this in a broader kind of simulation, which we've now done in this paper. Okay, sorry for going on so long about that, but hopefully that then convinces you that in a model toy system where you can create and break synchrony, there are real consequences for in-the-moment social interaction and lasting effects on behavior, particularly foraging and exploratory behavior in the pup. So just to summarize, I'm so sorry for going on for so long, but I think I hopefully shown you that there are some really interesting mechanisms by which neurosynchrony can be created, that it seems to be sensitive to the ostensive quality, the motivational quality of the signals that are being exchanged. It can be achieved through linear entrainment mechanisms like this flicker or the nursery rhymes, but that going ahead, we really need to be able to capture the nonlinear dynamics, because this is, I think, what really happens in real life. And it can affect not just in the moment behavior, but also longer term social decision-making and creating more stable states of cognition. Of course, we still don't know how this works in real life. All the systems I've shown you are really well-controlled lab experiments. So we really don't know how this works outside the lab. So that's still a mystery for us. And the other thing, which is really interesting, is, how plastic are these systems, right? do we have more or less propensity to synchronize with some people? And what changes that over time? Is that something we grow into and we grow out of? So these are some of the learnings that I have gleaned to date after working for nearly 10 years on the synchrony phenomena. And I hope you found that interesting. So it just remains for me to thank my wonderful research team, both in Singapore and in Cambridge, to thank all the funders, and to thank you very much for your attention. Thanks, everybody. I should also say, I promised Daniel I would advertise this. If anybody is interested in learning more about the work, I'm very happy to host you in my Singapore lab. And we are very open to hosting PhD students or visiting fellows. So please do let me know if you're interested in collaborating.
Wonderful.
I'm so glad you enjoyed it.
Thank you very much, Vicky, for such a richful and complex and comprehensive presentation. And along with Daniel's presentation, I think that it gives us a hint on how further we could go in our researchers with the collaboration with Professor Victoria broad skills of deepening the knowledge and make it even more comprehensive that we thought in the beginning of our studies, I think. I don't know what you think, Daniel. But a part of thanking both for their brilliant presentations, I would like to open the discussion with the audience. So if you have any comments, questions, provocations, please, I just would ask you to raise your hand and then present yourself, introduce yourself, and then we'll begin the discussion now. You could speak either in English or Portuguese, and we will translate to Professor Victoria. Okay, Julia. Please tell us who you are, because I know you, but they don't. Yes, but you are much more than your name. Yes, maybe you just say where you are a PhD student.
Investigating use of occupational radiation as an adjunctive treatment, adjunctive to the GPM treatment in borderline adolescents. GPM is it's a good psychiatric management developed by John Wenderson. It's a psychiatric management, generalistic approach, and way through my research, and I still have a whole way to go, but yeah. So to my question slash comment. I have a GPM to pregnant women with BPD, borderline personality disorder. And yesterday, we were discussing how she can advance in her research. Carla Sharp is also here, and I think she said to my colleague, like, look for Victoria's papers. She's been measuring. She's been working on measurements about the interpersonal hypersensibility that is presented in BPD. And I don't know if I have a question, but I've been thinking about the hypersensibility in the interpersonal model that is the base for GPM treatment. And I don't know, I guess I want to hear you, I want to hear more about how can we, how can you see the interpersonal hypersensitivity in, for example, BPD women, and how can it affect, because you said, you commented about depression, how can it affect this, the social and cognitive aspects of the development of the baby?
Yep.
And I would like to hear some, if you talk about BPD and what it can affect the development of the baby.
Okay, shall I take this one?
Please.
Thank you, Julie. That's a really great question, and I'm so glad so many of you in in this group and in other groups are interested in clinical applications, because I think this is where these theories and tools can have real utility. I don't work specifically with BPD, but we can talk more generally about disorders or breakdowns of the parent-child attunement process. And that is one of the reasons why I wanted to do the kind of modeling that we did here, because I think there's not a simple relationship that more synchrony is better or more contingency is better. It's not linear, right? Each of these different patterns of behavior will shift the cognitive states of the diet and the child in a different direction. And there's no, like, the more you go, the more it's no really simple relationships. It's not the case that, let's say we measure BPD and okay, they're not very contingent. We just need to get into more contingent and that's it. It's not as simple as that. What you need to be able to do is capture the state space, map the parameters, and then figure out where that sweet spot for that diet and that family should lie, and then bring them back to that. And I think this is one of the things that we're seeing in this hyperbola here. I didn't get a chance to explain this graph to you. But if I just go through that for you now. So this is the animal data, but we were simulating using our equation of state, what would happen to the system stability. So the y-axis is system stability, where lower is more stable. If you had different levels of contingency for the sync and desync situations. So it's very clear that when your diets have high contingency, which means they're going back and forth and they're very attuned, you can achieve really good levels of stability. That's what we're usually seeing in a very healthy system. When all the parameters are lined up in the right direction, you create very nice system stability. And when you break that with the desynchronization, then you lose all these benefits. The system is just a lot less stable. But the same thing happens when, even if you're technically synchronizing them, but there's no contingency in the behavior, you still don't get this nice stability, because all of this up here with low contingency is still non-stable. So that's one thing. But the other thing which is interesting is, what's on the x-axis here is the duration of the interactions. As I said, this is multidimensional, right? So it's not the case that more duration is better. The longer you spend in any behavior, the more stable you get. No, at some point it becomes diminishing returns, right? If you're too stuck, you stay too long, that creates inertia in the system, and then that actually has negative effects overall. So this is one of the ways why I think it's so important to model these state spaces, because there's not a simple answer here. I think we have to figure out in BPD what this looks like for the mom and the child. Are they pushed there, there, and then figure out where your sweet spot is. But then it could help you make more precise hypothesis-driven approaches to how you might want the interventions to push behavior in one direction or the other. Because already off the top of my head, I can tell you that there are some diets, let's say in anxiety, where we see hypersynchrony. That's just the mom is driving the child really hard in terms of the emotional reactivity, the stress, and the expressions are really intense. And so that is too strong a synchronizing influence on the child. It stresses the child out. So if the child cannot decouple, that's actually a bad thing for their development because they're getting this almost over stressful stimulation from the anxious mom. So that's an example of this kind of diminishing returns thing. So although I don't work with BPD, I would suspect there's a similar kind of calibration that we need to go through there to understand what their state space looks like and what is the optimal point that we want to bring them to. And you want your therapy to be able to target them to. In another piece of work, I have looked at the therapeutic alliance, which is a bit different, but in this case between an adult therapist and an adult client. And we did start to look at whether brain synchrony was a good measure of that. And there were some preliminary indications that could be the case. Unfortunately, that study was stopped because of COVID, and then my collaborator got too busy. But I have the protocols, I have the preliminary data, and if anyone wants to revive that, it's you know how to do it now. We can look at the therapeutic alliance with these hyperscanning methods. So it doesn't have to be parent and child, it can be therapist and client, or any other diet that you're interested in assessing this attunement parameter for.
Jill.
Yeah.
Who else? Thank you, Julia, for participating and for your answer as well. Yes, absolutely.
How can it affect mentalization process? The more synchronized, the more I don't know. I see a link to the mentalization process, development of this, but is it related to this synchronized this mom-baby synchronized process? I know it has some relation to the mirror
neuron system.
Yeah.
Yeah, that's a really great question, right? So this is the higher higher cognitive effects of synchronization. So in the mom-baby literature, because these children are so young, we don't give theory of mind tasks or mentalization tasks at this particular age. However, there is related evidence from studies on teacher-student synchronization because there's quite a robust body of work, and I actually just put out a review article on this, so I can send that to you if you're interested. We reviewed all the hyperscanning articles on teacher-student synchrony, and there's now quite a good convergence that the stronger the attunement between teacher and student, the better the learning process. And it comes back to what you're saying, because the shared knowledge construction, the shared representations seem to be more effectively built when there's good mental alignment. And those working in the adult literature suggest that that's related to very nice predictive capacities on both sides, that because you understand each other very well, you can positively predict what the other person means, how they're going to complete their sentences, what the overarching knowledge structure you're trying to create is. And that has been reflected in the synchrony of the brain signals. So I think in the adult literature, there are hints at that, but in our developmental field, we haven't, or at least I haven't, maybe Pascal has, but I haven't looked specifically at mentalization because of the age range that I'm working with. But we have looked at self-regulation and other types of skills, and we know that synchrony, especially between mother and child at the really early age, is positively predictive of the child's self-regulation, emotional control skills later on. And that is related then to developing executive function. So you can see how you would have knock-on rolling effects. Even if it's not direct, it may have indirect effects later in cognition development. But certainly in the adult side, there's a good suggestion that may be one of the ways in which synchrony does benefit knowledge acquisition and knowledge co-construction.
Who else? Leopoldo. Claro, claro.
First, I would like to agradecer a você pela apresentação.
He thanks you for your presentation.
my first feeling is that que eu estou na idade da pedra, comparado às pesquisas que você apresentou sobre a sincronia do bebê e nos estudos de neurociência. Né?
He feels that he's in the Stone Age as compared to your research on synchrony and the neurosciences of the bond between mother and child.
It's real. And, but, eu tenho feito estudos sobre teorias psicanalíticas do desenvolvimento.
He has been studying psychoanalytic theories on development.
E sempre que eu me aproximo dos estudos de neurociência, eu me pergunto qual é a teoria de desenvolvimento de fundo utilizada para fazer essas pesquisas.
And when he approaches neurosciences studies, he asks himself, what is the theory on development which underpins such studies?
Porque cada teoria do desenvolvimento teria, por sua vez, modelo ontológico de homem.
Since each theory on development would have an ontologic framework on how human beings develop,
diferentes concepções sobre o que é a mente,
different conceptions on mind,
ou outros termos como vida psíquica, pensamento, pensamento, em algumas teorias, pensamento e mind não são a mesma coisa, psique e mind não são a mesma coisa.
In some theories, psyche and mind do not overlap, nor thoughts.
E para não me estender muito, nós teríamos talvez de ter, me pergunto qual é a teoria de comunicação de fundo para explicar a sincronia, porque nós podemos ver sincronias objetivamente identificáveis por esses instrumentos.
So, what would be the communication theory which underpins the synchrony process?
E a interpretação dos dados objetivos da sincronia em cada teoria.
The interpretation of the data on synchrony in each theory
pode ser diferente.
Might be different.
E isto significa não só sentido diferente.
And this means not only a different meaning,
mas pode estar se referindo a fenômenos, realidades empíricas diferentes.
But also to different empirical realities, different phenomena.
Okay.
Shall I try?
Please.
First of all, that was an incredibly sophisticated and very difficult question, but I will do my best to answer it. So how do we do this? Do I answer and then you translate?
I'm going to translate.
Okay. So first, can we compliment him on the fantastic question?
Yes. Ela te elogiou pela tua pergunta tão sofisticada e difícil que ela vai tentar responder.
No, it's fantastic because, I really don't, I think the classic theories are so powerful it's primarily about how you understand knowledge and what is your framework for understanding the basis of is being constructed out of your data, what knowledge are you gaining from this way of analyzing the data. So I would say in a nutshell.
Você que eu traduzo? Então, sorry.
Yeah.
So, então ela falou que as teorias clássicas são muito poderosas e elas influenciam a maneira como a gente interpreta nossos dados.
Okay, so I think what we're learning from my work is the specific constraints, neural constraints, which dictate how a social partner influences the child's brain development, the neural constraints.
Então, ela está preocupada em entender como algumas diretrizes influenciam o papel da mãe no desenvolvimento da criança no que diz respeito justamente a esse processo de sincronia e desincronia.
So a powerful social partner, which has a strong influence on the child, we will lock onto the neural signals and then train the neural signals of the child very effectively.
Então, um adulto que tem uma capacidade poderosa de engajar a atenção da criança, a sua sincronia, num conjunto de pistas, né, ela não falou isso, mas eu vou chamar assim, porque a ideia me vem dessa maneira e eu acho útil. Âncoras de pistas sociais definidas.
And that will be dictated by the structure of the child's neural system. So its oscillation rates, the temporal patterning that will be effective in driving it, the duration and the contingency, all of that will create either strong or less strong entrainment. And that will then have effects on the child's brain development. So it's the match between the input and the child's receiver system. That's what we're really measuring by this synchrony or contingency technique.
Então, o que ela estuda é como a atitude da pessoa que está em posição de cuidar, da pessoa adulta, nessas âncoras de pistas sociais, vai ao encontro da tendência do bebê, da tendência neurofisiológica do bebê, de maneira a impactar essas variáveis, né? Oscilação, sincronia, intensidade, temporalidade, no engajamento da dupla e seus efeitos para o desenvolvimento da criança.
And it also means we can predict when you change the type of input or when it breaks down, you can then predict the consequences, atypical consequences on child's development.
Então, quando você manipula essa âncora socio-interacional, você tem como predizer o seu potencial efeito sobre o desenvolvimento da criança, seja quando você manipula a intensidade, seja quando você trabalha numa perspectiva binária, por exemplo, de com e sem aquela variável.
So in a nutshell, we're studying the interface between the two systems, the constraints of that interface.
Então, para resumir, você está trabalhando na interface entre essas dimensões.
So I know it's not as sophisticated as, a psychoanalytic view of, development of self and self-concept, but it does describe how external influences impact the child's kind of basic brain developmental potential. And that's what we can contribute.
Então, ela sabe que do ponto de vista psicanalítico não é uma resposta sofisticada, mas ela está focando numa área bastante específica, né, que é como se dão os mecanismos, tanto neurológicos como das suas consequências, né, assim, modulação da sincronicidade e seus efeitos no desenvolvimento da criança em algumas variáveis, do ponto de vista da sua... Ela não falou isso, mas eu estou explicando, né, da sua predictibilidade.
Na verdade, eu achei sua resposta bem sofisticada.
He found your answer very sophisticated.
Porque se aplicássemos talvez esse mesmo tipo de estudo que você faz para dois adultos conversando em psicoterapia, ou
If we applied such study to two adults in a psychotherapeutic setting,
Uhum. E mesmo, por exemplo, na nossa própria comunicação,
and even in our bond, our communication,
talvez fosse possível
maybe it would be possible
identificar o que um psicanalista chamaria de uma comunicação profunda, inconsciente,
maybe it would be possible to identify what a psychoanalyst would call an unconscious communication
para além da informação específica que está sendo colocada.
beyond the specific information which is conveyed,
Mas em função da sincronia afetiva,
but as a function of the affective synchronicity.
e isto seria uma maneira, acho que muito sofisticada
It would be a very sophisticated manner of
e atual
and
de,
current
de identificar a própria matéria da amizade, do amor, da comunicação,
to identify the matter proper of love, friendship, communication.
que não são processos mentais. Que não são processos mentais.
Which are not mental processes.
Afetivos processos.
Yeah, absolutely. I think there is huge potential to do exactly what you said there, which is go beyond the perceptual input, the semantics, the syntax, the actual cues, to look at the non-tangibles, the emotional states, the sense of being, the shared sense of we understand each other, the connection. Yeah, I think that's really the potential that we can push towards. Now, I don't have the skills to know whether we're measuring the right thing. So I think we need to collaborate with people like you to tell us what we should be benchmarking against, to know which part of the neurosignal is telling us about these affective qualities. But I think there's great potential to do that.
Ela concorda e ela vê um potencial muito promissor da articulação da dimensão do afeto nas pesquisas dela, o que não é o objetivo primário.
Good idea. It sounds like there's good interest already
Yeah,
for using
yeah.
these techniques for your questions. Yeah. And by the way, congratulations on very effective translation. At some point, I think like you can give my talk.
Not at all. Thank you very much. You're so kind. Who else? Bartolomeo, please go ahead. He asking Professor Daniel. You found a socioeconomic variable as the most prominent risk factor in the Germina cohort. Even if Germina is not a representative sample, would Daniel think that this might have something to do with our socioeconomic situation as a nation?
So how do I proceed now? English and Portuguese?
You could translate yourself.
Yeah, I can. Just kidding. Okay, so the short answer is probably yes. Probably it must be related to inequalities. And some other thing that it's interesting is that most of the women were middle to upper class. So, so what our data is showing is that maybe there's also differences in terms of income, even when you consider middle class to the upper class, not only the only poor to the middle and middle to upper. Eu acho que a resposta é basicamente sim, e eu acho que essa questão da renda, que é um dos principais variáveis, né, que estavam nessa dimensão, mostra que entre a classe média e as classes mais ricas pode ser que tenha uma diferença bem importante mesmo, assim, que a gente costuma jogar tudo no mesmo saco, assim, talvez, né, quem é classe média, quem é muito rico, parece que tanto faz, porque todo mundo tem bastante dinheiro, mas talvez não seja tão simples. Então, na corte do Gemini aparece um pouco isso. However, it must be noted that the socioeconomic and social demographic variables in this model, they were not the we like merged all of them together and did a feature selection, so it's not that fine-grained. We didn't evaluate variable by variable. To understand a little bit more about the causality of each variable, we need different models. So here is more like brute force, for instance, because we are just throwing variables and see what the models come up with. E aí, acho que para finalizar, esse modelo ele é mais bruto, assim, por assim dizer. A gente joga um monte de variáveis em cada domínio que eu mostrei e a gente vê o que aparece. Então, eu não tenho tanto detalhe assim para falar, essa variável é a renda, é o ABEP, é a classe social. Então, a gente precisaria de modelos talvez um pouco diferentes se a gente quiser entender realmente no detalhe, assim, se era mais a renda, se era mais o sexo, né? Porque ali entra várias questões. Sexo, idade da mãe, idade do bebê, entra várias questões, assim. Mas com certeza a renda é um dos fatores mais importantes. E aí fica até como anedota aqui, na Germina a gente tem pessoas que ganham mais de 100 mil, assim, tipo, são outliers, assim, mas é curioso, né? A gente estava limpando os dados e falou, nossa, deve estar errado isso, né? E a gente foi confirmar e era verdade, assim. I was just talking about the one of specific family in our cohort that the income was so high that I thought it was wrong. Yeah, too many zeros, but then we confirmed later it was actually true.
Grover, would you like to chime in? Você gostaria de comentar a esse respeito? Aham. Uhum.
Aliás, até aproveitar para traduzir e comentar aqui, o Grover, ele é do IME, né? Trabalha com o professor André Fujita, tá desde o começo do Germina com a gente e sempre trabalhou muito assim nas análises, super parceiro aí. As análises que eu mostrei foram fortemente produzidas por ele, então super valeu aí. Por isso, mas basicamente para traduzir a resposta é que o modelo ele não preocupa tanto no nível do detalhe, né? A gente seleciona a variável, a gente pode saber, né? A gente consegue ver depois qual variável que foi selecionada, mas é difícil saber o tamanho do efeito, assim, será que a renda é um efeito maior do que o sexo? É um pouco difícil mesmo, então acho que confirma um pouco do que eu tinha dito.
May I
Yes.
ask you something? Okay. So, along your presentation, I've been constructing many articulations of your data, between your data and some neuroscientific theories. So I passed along affective neurosciences, Jaak Panksepp, Mark Solms, and also computational neurosciences, the work of Carl Friston, Robin Carhart Harris, and also the more recent work of Georg Nordhoff on spatial temporal neurosciences. And they all highlight different aspects of both the central neurosystem and also its outcome, its expression on functions which regard the mind. So, considering all this, I would like to ask you something related to what Leopoldo did, but in a different way. Neuroscientifically speaking, what would you say is the role of affect in not all, but some of your results? And considering this, what do you think is the function of moments of non-synchrony between the dyad, I mean the mother and the infant specifically? And if you agreed to add something else, what would be the role of the default mode network and the brainstem?
Wow, your questions are even harder. I think, I think we may need to have lunch to answer these questions, but they are fantastic. So, affect. What comes most immediately to mind is the reward signal, and how that either is interpreted as reinforcing the current behavior and motivating its repetition, its learning, etc., its reinforcing qualities, or for it to be an aversive stimulus. So I think in my experiments, and we've got a few where we manipulate emotion like that, we've primarily treated emotion in that sort of context as both motivational / reward signal. And I think that's one way of seeing it, that when we look at synchrony, you get the increase in synchrony, partly because the child and the mother are enjoying the interaction, they're sharing positive affect, and so because they want to continue repeating the behaviors that have been generating that, you reinforce that coupling strength. So that's one explanation, which is an indicator of the potential of the system to keep persisting in its current synchronous state because it's very rewarding for both individuals. So that's one dimension. And of course, it can also give an error signal for the system, because when things are very positive and you're happy, that's great. But if affect suddenly changes, you know something has changed and you need to correct. And so that may then temporarily cause a break in synchrony, because both parties will then withdraw a little bit from what they're doing and readjust their behavior. So I think it's quite a useful way of predicting whether a system is likely to persist in its current state and maintain the high levels of synchrony, or whether there's likely to be a break or there's caused a break because of the change in the effect. And when we look at the effect sizes, because we have different domains and we measure the relative effect sizes on synchrony, emotion has one of the largest effect sizes, and it is related to longer periods of coupling. So the gaze effects are more volatile and shorter lived. The auditory ones are also quite short lived, but the emotion effects tend to be really powerful, actually. So I think this is why your question is a really great one, because it may be one of the most fundamental domains that is driving or helping us understand the dynamics, the synchrony dynamics within the pair. But that is from my perspective of looking at motivation and reward signals in the dopamine system and the oxytocinergic system. Maybe there are other systems that are involved, which are related to synchrony that play other roles. Certainly things like, evaluating threat or, emotional reactivity, all that could play into that as well. But for me, just looking at healthy diets, I think that is one important role that we need to understand as a flag for synchrony levels within the diet. So that's the first question. What was your second question again?
I think that my second question was...
That was his third question.
Yes. No, the first would be what role of the desynchronization
Yes, correct. That's right.
periods between the diet.
So I kind of touched on that already, because like I started at the beginning, desynchrony states are transient. So some desynchronization is just spontaneous because systems are vulnerable to chaos and entropy. And over time, if there's no impetus to keep them synchronized, they will decay. So some desynchrony is a natural state because whatever is generating synchrony, that forces just, dissipate it. And so you will naturally desynchronize, right? But I think there are also cues for desynchrony. And that's where I think the negative emotion can be quite helpful, because I think it triggers a need for the system to recalibrate. So usually when there's a negative signal, the system will readjust itself. So I think, and we study this with things like the still face paradigm.
Electronic.
Yeah, still face paradigm. So there's a negative affect, although it's not, I'm depressed, it's non-responsive. And that's viewed by the child as negative. And that, across our work and other people's work, seems to trigger desynchronization because you break the contingency. There's no reinforcing behavior anymore. And it causes the system to desynchronize that way because there's no kind of back and forth anymore. So I think that happens in pathological situations, in situations where the system needs to recalibrate. And it's a useful signal. It tells the system that something needs to change or something has changed. So I think that's its natural role, but also, as we discussed, I think when we answered Julia's question, in some cases where synchrony is pathological, then desynchrony might be more adaptive for the child. So that's where, the spontaneous high levels of coupling because of high emotion or a driving force that's very strong, which is not positive, the child needs to learn to desynchronize. So they become the non-responsive partner. And so I think in all these cases, desynchrony is meaningful. Either it means the lack of a driving force or it means the system needs to recalibrate in some way because there's negative feedback. So I think both synchrony and desynchrony are equally meaningful. I don't think they are, one is good and one is bad. I think they are useful ways of interpreting information and responding to environmental input. For systems. And systems need to desynchronize because synchrony is a very locked-in state. And potentially energetically, it's also quite an expensive state because you have to keep
absolutely
synchronizing. So I think in fact, if you ask me, most of the time, we're more desynchronized than synchronized. Synchrony seems to be a more privileged state that we achieve maybe for communication purposes when we need high understanding, high attention, high focus. We create high synchrony. But otherwise, systems more likely disentangle and become more entropic over time.
I might have an alternative hypothesis.
Please, yes.
First of all, you have just provided an evidence on unconscious communication because I had electronic in my mind.
Oh, there we go.
Yes.
And then I said it.
See, and he has trained a group of researchers at the University of Buenos Aires, whom I have been working with, and they have built a protocol to investigate synchrony and desynchrony between mothers and infants, and to assess the level of the symbolization of their interaction while playing. We have applied this to our research in a follow-up step of our study, and it's very interesting. So, what they found is that this synchronization is valuable not only to serve as an alternative to excessive synchronization processes, and not only as a reaction to negative affects, but also in other situations. It kind of comes along with synchronization processes. So, what would you think of the possibility of the need of desynchronization processes as to convey working memory and declarative long-term memories to convey them into long process non-declarative ones?
Wow.
What I'm saying is that one thing is being related to the connection and feeling and processing, learning, and another thing is building inside of the internalizing the own version of these experiences.
I see what you mean. And actually, this ties into your third question about the
Yes,
default
absolutely.
network.
Yeah.
Because I was about to answer in the same way. You know, we have states which focus on our internal state. You're kind of conscious of your own cognitive state. And it means that you're slightly closed off to environmental stimulation. And the default mode network is the strongest when you're ruminating, when you focus on your internal state. When you're talking to people, that kind of dissipates because you're focusing on the external environment. And of course, in synchronization, we measure the externally focused state. But in order to internalize information, you have to grab that and somehow encode that and change your mental representations, right? So I think synchrony is that first step, which tells us how good a grip you have or how strong your grip is on the external stimulus that you can bring in. But then something is missing. We don't really understand then how that then gets locked in to your internal system. And maybe that's where we need to look at the external and internal default network integration of those two coupling systems. We've started to look at that in our latest experiments where we model both. We have the child's internal connectivity and we have the cross-brain connectivity. And we do see that they are separable in function, but they kind of do have a causal role. One causes changes in the other. So we're starting to get there, but we're not there yet. And we've looked at that in the context of a gaze paradigm, where again, it was speakers talking to babies, looking at them, blah, blah, blah. And we again saw these different roles. The cross-brain signal was very powerful in gating the capture of information, but then the internal connectivity with the child was then responsible for locking that into the learning of the child. So I think that is the next frontier. You put your finger on it. But yeah, we don't understand that nexus very well. That facing out, facing in, facing out, facing in dynamic is something we still need to understand better.
Thank you. Thank you very much.
You guys are amazing. I should spend more time here.
We would be delighted to do this. Who else? So would you think we had enough?
We continue over a drink.
Would you like to add something, Daniel or Vicky?
No, I don't think I have much to add to what you were discussing. Yeah, I just would like to thank you for organizing this. I would call it an event, right? I think it's an event. So very nice to be here. Very nice to collaborate with Vicky. Since we met in the beginning of our cohort, it was always very nice in terms of discussions. And then we didn't mention this that much, but you also collaborated with us in a subsample of the Germina cohort, where we did a dyadic EEG. So we have the dyadic EEG data that we started to analyze, and we'll be publishing very soon. So it was very good, and we are planning much more future projects, right? So we and we have a lot planned at the Institute of Psychiatry, and also here at the Institute of Psychology. We can think about a few more projects, right? So, but we do need to have more people. That's why you mentioned at the end of your lecture that as we start to contemplate these complexities of behavior, and especially dyadic behavior, we need more complex frameworks of analysis, more complex frameworks of theories also, just like Leopoldo was mentioning. So we need to train people. We need capacity building. So because science will get more and more complex as we go along. It's not get any more simple. So Vicky was very generous in offering to us to host students in the very, very near future, in fact. So we are mostly interested in students that can learn these models, some of the models that Vicky showed, especially the nonlinear dynamics that are derived from physics and mathematics. We are very much interested in people that could be trained in these models, but not only that, but also how to run these experiments. So acquire the EEGs. For instance, the dyadic EEG experiments, they are very different, very specific from traditional EEG that sometimes we are just doing like resting states and stuff like that. It's more complex than that. So we need people that can come back to Brazil and run some of these experiments. So it will be very, very good to build this knowledge to do more and more studies in the early childhood development field. So yeah, I think that's it. Yeah.
Nossa discussão é muito estimulante e
Our discussion is very exciting.
Eu queria comentar só uma coisa de talvez um campo, um período de pesquisa.
He would like to add something about a research field.
Pensando talvez numa futura colaboração entre nós.
Thinking of a future collaboration among us.
Eu percebi que seu estudo começa com crianças de 3 meses.
I realized that your studies begin with children of 3 months of age.
Algumas teorias do desenvolvimento
Some theories on development
consideram que o nascimento da noção de tempo e de espaço no bebê
consider that the uprising of the notion of time and space in the child
tem origem na relação primitiva mãe-bebê nos 4 primeiros meses.
Derive from the early interaction between mother and infant along the first 4 months of life.
Dando salto, por exemplo, numa perspectiva de uma psiquiatria fenomenológica.
So, considering phenomenologic psychiatry,
os grandes problemas, os problemas fundamentais dos psicóticos,
the fundamental problems, issues of the psychotic people,
seria a não fundação ou não aquisição de uma noção de temporalidade, especialidade e espacialidade.
Would be the difficulty in building the notion of spatial and temporal features.
Logo, uma pesquisa que pudesse analisar a sincronicidade mãe-bebê do nascimento até o 3º mês.
So a study which could I'm sorry. So I'm thinking of the content, what he's saying. So a research that could assess the synchronicity, the temporal and spatial synchronicity between mother and infant along the 3 first months of life,
Especialmente no fato ou fenômeno da amamentação, que seria um fenômeno especial para verificar tal sincronia,
mostly during breastfeeding,
e associado com alguma maneira de verificar se o bebê está adquirindo uma noção de temporalidade,
associated to some way of assessing if the baby is acquiring any capacity of interacting with the features of spatial and temporal synchronicity.
poderia oferecer um conhecimento efetivo, poderoso, para ser utilizado nas políticas de saúde pública mundiais.
Could bring an effective and powerful knowledge to contribute to public policy in development.
I think that's a fantastic, take on the power of this research. I am very interested in this model building that you've mentioned. So how does the child acquire their fundamental assumptions about the physical world and the emotional world and set up their expectations? And that's based on the received input from the mother, what you talked about, and training from the social side, but then internalizing that into your mental model. So I think that is exactly tapping into that bit. How do we measure the resulting mental model of the child as a result of these inputs? So I'm very excited if you can give me paradigms and ways to benchmark what I'm measuring. I can definitely create the neural measurements and the modeling, these dynamic systems modeling, to pick apart healthy and not healthy development of those mental models. I think that's a really great idea for how we could partner in this together.
Very good. Ela concordou com o que você falou, a importância do que você falou, inclusive a articulação entre isso e o que a gente estava discutindo antes, e ela se mostra aberta para, se a gente conseguir formular isso em termos de um paradigma, ela propor quais seriam os modelos de avaliação neurocientíficas dessa questão.
Yes, I hope so. Yeah. I think it's exactly
Hopefully, in the near future.
Yeah.
Okay, so I would like to thank all of you for coming, and Vicky for being so generous and so didactic in presenting all these comprehensive and sophisticated knowledge as a result of her endlessly capacity of working and researching, and also for being here and collaborating with us and being open to further collaborations, and also Danielle for making it possible to Vicky to come to São Paulo along with the whole staff of the Institute of Psychiatry, and Professor Guilherme Polanczyk, Adriana Argeu, Monique, I forgot her surname, Monique Teixeira, and a special thanks to Elizabeth Shepard, Lizzie, and I'm sure she would be participating in our discussion, and that it will happen in a near future. So it's very good to have Lizzie with us, and she will collaborate with her very richful and exciting way of considering all of this. And we have a bright future to walk along in our partnership. And thank you very much. Yes, yes, yes.
Just a second. I forgot to mention about Lizzie during my presentation. So the paper that I mentioned, I have shared first authorship with her. She was fundamental since the beginning of the Germina cohort because, as you may suspect by now, I'm not a specialist in early childhood development. I'm most interested in mental disorders. I came from a child adolescent psychiatry background. So she was fundamental in thinking about the study, what measures we would use. All the EEG paradigms that we used came from her knowledge. And yeah, we've been collaborating so much for the past few years since she arrived in Brazil, in fact, during the Primeiros Laços project. So unfortunately, she couldn't be here, but yeah, I would like to document that I really thank her for all the help with all the analyses, all the papers, and of course, much more in the future also.
I would also like to mention that she has a very sophisticated trial. It's a randomly controlled trial on early risks for ADHD and autism, which is called, her study is called Florea, and it's a very interesting design of research, and you can access the protocol, which is already registered, and also in the social media. They have Instagram and a website, and it's a very interesting research which doesn't have preliminary results yet, but it's very promising. So we're very happy to host her research here in our institute, and since its beginning it has been hosted in the Institute of Psychiatry, but since she became a professor here, we will give continuity to her works, always in a partnership with the Institute of Psychiatry. It's a very richful opportunity for all of us. So thank you, Lizzie. I hope you're watching us right now, or if not, maybe you could see the video registering of this event. And thank you all again.
Alô, som. Alô, som.